you, everyone, for joining today's webinar on datasets through the looking glass. My name is Mamunur Rahaman, and I'm a PhD candidate at the University of New South Wales in Sydney, Australia. Today, I'll be presenting advancing computational pathology, multimodal datasets, and deep learning insights. I'll share how we can integrate deep learning with multimodal datasets, including histopathology images, spatial transcriptomics, and clinical trial data to improve cancer diagnosis, prognostication, and treatment personalization. So let me first briefly introduce my academic background and research impact. I completed my bachelor in electrical and electronic engineering from BRAC University in Bangladesh. I then pursued masters in biomedical engineering at Northeastern University in China, where I focused on deep learning in computational pathology. Currently, I'm a PhD candidate at UNSW Sydney, focusing on AI and machine learning, machine vision for computational pathology. During my PhD, I was also a visiting fellow at Georgia Institute of Technology and Emory University in the United States, where I collaborated with Professor Anant Madhavushi's group on advanced AI techniques for cancer research. In terms of research impact, I have contributed over 40 peer-reviewed papers with more than 3,200 citations on Google Scholar. 4 of my papers have been recognized as ESI highly cited papers, placing them in the top 1% in the engineering field globally. I have been also fortunate to receive approximately 300,000 US dollar in major funding and awards throughout my academic journey. So what exactly is computational pathology? In essence, it's about leveraging AI to assist, augment, and advance pathology and oncology. At the center of everything is whole slide image, a digitalized tissue sample in a gigapixel scale. So the slide contains enormous information that enable us to accomplish 4 distinct but interconnected clinical tasks. First, detection and segmentation. We can automatically identify the region of interest, tumor versus the normal tissue. So this automates what pathologists traditionally do manually. Second, molecular profile prediction. AI can predict gene expression patterns, genetic mutation, molecular subtype directly from the tissue morphology. Third, prognostic and risk stratification. We can classify patients into high risk versus low risk groups and do survival analysis. This helps clinicians make informed treatment decision based on predicted patient outcome. And fourth, clinical outcome prediction and classification. From image patches, we extract the features to predict the clinical outcomes, benign versus malignant, tumor grade, treatment response, and other diagnostic classification. The promise of computational pathology is transformative. We can provide faster, more objective diagnosis, risk stratification, predict molecular features, and ultimately guide personalized therapy decisions. However, realizing this potential require overcoming several significant technical and clinical challenges, so which is exactly what my research addresses. So now, while the computational pathology is promising, the data itself is extremely complex. You can see in the image, a single whole slide image can be in a gigapixel scale with the width approximately 143,000 pixels and the height is 84,000 pixels. This image have multiple zoom levels. So we can look at the entire tissue or zoom in to see individual cell. But size isn't the only challenge. We also face significant color and staining variability across different laboratories and scanners. There are imaging artifacts, and critically, there are intra-tumor heterogeneity, which means different region of the same tumor can look different and have different molecular profiles. So all of these factors mean that we need a specialized pre-processing and post-processing pipeline. Standard computer vision approaches often fail because that are not designed for medical images of the scale and complexity. So how do we actually build AI for these huge images? We use a three-step approach. So first we create patches of this huge whole slide image. So typically 2 to 4 by 2 to 4. In step 2, we use AI to extract the key features from each patch. So the neural network can extract patterns from those images like cell shapes, tissue structure, and color. In step 3, we combine information from all those patches to make a final prediction for the whole slide, whether it's diagnosis, prognosis, or molecular features. So today I'll be giving a brief overview of 5 interconnected projects that build on one another to advance computational pathology. First, we build the foundation with HistopathAI, a robust generalizable classification model that handles the gigapixel whole slide images across diverse dataset. Second, we create interpretable biomarkers for head and neck cancer. Third, we bridge histology and genomics with BrST-Net. Fourth, we achieve clinical impact through ST-DoxPCa using virtual spatial transcriptomics. We guide chemotherapy decision in prostate cancer. Finally, we solve the data limitation problem with MR-PHE, which is a zero-shot learning framework. So this project directly addressed 3 key challenges: weak generalization, missing molecular insight, and data scarcity. By delivering domain robust features, virtual spatial transcriptomics leading to therapy guidance and zero-shot classification. Project 1 is HistopathAI, so which establish a robust foundation for the histopathology image classification. The problem we are addressing is that traditional cross-entropy trained classifier tend to overfit to specific domain. They work well on the data that they are trained on, but fail when we tested on new data set from different hospital or scanner. So our solution involves 2 key innovations. First, supervised contrastive learning in the pre-training stage, which creates a cleaner and discriminative feature space, as you can see in the TSNE visualization. With supervised contrastive learning, get much tighter, more separate cluster compared to the standard training. Second, we use hybrid deep feature fusion during fine-tuning, which combines feature from multiple layers of the network to capture both fine-grained and high-level semantic information. So this is the technical framework in more detail. So it has 2 key components. First is supervised contrastive learning. Second is hybrid deep feature fusion. And our training strategy is 2-stage. In stage 1, pre-train the entire network using the supervised contrastive loss. And in stage 2, fine-tunes using standard cross-entropy. So to demonstrate the generalization, we have evaluated our framework across 8 diverse public dataset and 1 in-house dataset across 3 different cancer types: breast, colorectal, and gastric cancer. And in this slide, you can see the key results across 8 dataset in 3 different cancer types. We obtained the state-of-the-art accuracy compared to the existing studies. So to summarize, Project 1, the key contribution is establishing a robust and generalizable framework for the histopathology classification. Now, having established this strong foundation, we can move beyond simple classification to clinically actionable insights. The next challenge is to apply this foundation to build an interpretable AI-driven biomarker for predicting the outcome in head and neck cancer, which is the project 2. The project 2 tackles clinical problem in the head and neck cancer. The challenge is threefold. First, it's highly variable, making patient outcome hard to predict. Second, immunotherapy works only 15 to 20% of the patient. And third is current biomarker like PDL1 are unreliable at predicting who will benefit. So our solution focuses on the tumor microenvironment. So using routine H&E slide, we quantify three key features: dentil, which measures the immune cell density and organization; spatial, captures the spatial layout of immune cluster; and collagen characterizes the stromal architecture. Together, these three pillars create an interpretable, comprehensive view of how tumor interact within its microenvironment. So here you can see these three pillar biomarkers. So the top row for dental and spatial, we start by detecting all cell nuclei and identifying the immune cell. Then we build connections between nearby cells and create a cell subgraph. And finally, we group cells into clusters to understand their spatial organization. In the bottom row for collagen features, we identify stromal region in the tissue, then detect and analyze the orientation of collagen fiber, shown here in blue. So this slide illustrates the multi-cohort validation workflow for this HNSCC biomarker. We developed and trained the model on two institutional cohort, University Hospital and Emory. This slide summarizes the prognostic power of our combined spatial, dental, and collagen model. So the result demonstrate that integrating all three feature set yield the strongest and more reliable prognostic signature. For immunotherapy response prediction, we achieved very high accuracy, and the ROC card demonstrate the performance across different classifier using the 5-fold cross validation. So to summarize, project 2, we have developed interpretable multimodal biomarker for the head and neck cancer. This provides scalable objective risk stratification and immunotherapy response prediction. Now we want to see whether we could develop a molecular and gene expression signal directly from H&E. So project 3 tackles spatial transcriptomics. So we know that spatial transcriptomics map gene expression in tissue, but it's too expensive and complex for routine use. Our goal is to predict the spatial gene expression directly from H&E slides to generate virtual spatial transcriptomics. So here in the image, you can see the colored spot show the predicted gene expression from healthy, which is green, and cancer, red. So this would make molecular profiling accessible to every patient. This diagram show our key innovation for the BrST-Net framework that directly predict hundreds of genes from the routine H&E slides. So there's a issue of overfitting when we tend to predict around 250 genes. To overcome this, we train an auxiliary network alongside the main gene prediction network. So it acts as a regularization and forcing the model to learn meaningful features that generalize better. So the results are remarkable. Our model could predict 237 genes out of 250 genes with a statistically significant classification correlation. So in the figure, you can see how accurately our prediction captures the spatial patterns. The color matches closely between the column C and D, the ground truth and the prediction. This demonstrate that BrST-Net is learning real biological relationship between the tissue and the gene expression. To summarize the project 3, BrST-Net establish a powerful and validated method for creating virtual spatial transcriptomic profile from standard H&E slides. So this leads to the next project on prostate cancer, where I apply this virtual spatial transcriptomic technology to build a predictive biomarker that guides chemotherapy decision in prostate cancer. So project 4 overcomes a major clinical dilemma in prostate cancer treatment. For men with high-risk prostate cancer, adding docetaxel chemotherapy to their treatment can improve survival, which is a good news, but the problem is it doesn't work for everyone. Currently, there is no reliable biomarker to predict who will actually benefit from docetaxel. This means many men are receiving this toxic and deliberating treatment with absolutely no therapeutic benefit. So they suffer from this toxicity without getting any benefit. Our goal is to develop a model, ST-DoxPCa, by using the virtual spatial transcriptomic to create a predictive biomarker that can guide this critical treatment decision. This slide shows our complete study design. So it has 3 key steps. First, we train a virtual spatial transcriptomics model using the HIST-1K prostate cancer spatial transcriptomics dataset. Second, we apply this trained model to extract predicted gene expression features from 2 independent cohorts, the CHAARTED clinical trial for metastatic prostate cancer and the Cleveland Clinic prostatectomy cohort for localized disease. We use the CHAARTED data to build a treatment response model and the Cleveland Clinic data to build a prognostic model. Third, we perform external validation by applying the Cleveland Clinic prognostic model directly to the RTOG 0521 clinical trial. The survival curves show our biomarker successfully stratify patient in both CHAARTED and RTOG 0521. In high-risk patient, docetaxel provides significant survival benefits. In low-risk patient, there's no benefit, suggesting they can safely avoid chemotherapy toxicity. These demonstrate how virtual spatial transcriptomic can guide real treatment decision across multiple clinical trial dataset. So first, let me show the gene expression prediction performance. This histogram show the distribution of Pearson correlation coefficient for our 208 predicted gene. The majority of the genes show positive correlation between the predicted and the ground truth, which with many as achieving the correlation above 0.4. For the 208 genes, we have from this 208 genes, we have selected 26 genes based on the prostate cancer, and the prediction is even better.
Excuse me, Mamu.
Here's the
if you can finish in the next few minutes so that we stay in time.
Yeah.
Thank you.
So yeah, sure. So I'll finish by 5 minutes. So here's the virtual visual demonstration of our virtual spatial transcriptomics, and the left is the H&E, middle actual gene expression, and the right is the prediction. And you can see the prediction is closely matched with the ground truth. Here is the key validation result from 2 landmark trials. The survival curves show it successfully stratified patient in both RTOG 0521 and CHAARTED cohorts. So yeah, we also validated ST-DoxPCa in the Cleveland Clinic Foundation cohort for the biochemical recurrence, and we found statistical significant result. Um, so this is the model, what model is actually learning at the molecular level. So you can see the whole slide image shows the prostate sample from the CHAARTED trial, and the heat map visualize the model's prediction expression for the gene KLK4 across the entire slide. The warm color indicate the higher predicted expression level, and this visualization also demonstrate that our model isn't just making the random prediction, it's identifying specific morphological pattern that correlates with the gene expression. To summarize Project 4, we have translated novel AI technology, virtual spatial transcriptomics, into clinically actionable predictive biomarker validated on 2 major randomized clinical trials. In the next project, we try to tackle the fundamental challenge about the data scarcity. So in this project 5, we try to see if the AI model um, can do the classification without the label trained data. So here's the challenge, the same image with 3 different text prompt describing benign. The heat map shows where the model focuses, and they are completely different. So subtle word change shift attention to different region. So we have proposed MR-PHE, so which mimic the pathologist, actually how pathology actually examine the slides. Um, so we extract patches from 3 different scale, then we um, implemented advanced prompt engineering, and then we implemented the hybrid embedding strategy. So the result is promising. So first, our model beats all the zero-shot learning method by 2 to 18% across 6 datasets. And second, we often exit fully supervised model despite using the zero training data. And third, outperforms pathology foundation model like COMS and FLIP. So to summarize, Project 5, it represents a paradigm shift. So it is the first framework to achieve the reliable zero-shot classification accuracy in histopathology across multiple data sets and tasks. So to summarize this journey, collectively, we have developed 5 projects from a comprehensive set of AI-driven tool that advance computational pathology toward precision oncology. They make cancer diagnosis faster, more objective, and more accessible worldwide. Looking ahead, our next step is to validate this AI tool in prospective multi-cancer clinical trials, demonstrating their real-world impact on patient care. Simultaneously, we will integrate emerging data modalities like spatial proteomics, radiomics to create more rich multimodal biomarkers. We aim to pre-train large pathology-specific foundation model to further improve the zero-shot learning capabilities. We also plan to embed this tool into digital pathology workflow for real-world decision system. Finally, we are committed to addressing health disparities by developing and validating our model across diverse population and low resource setting, ensuring equitable access to this advanced technology. So thanks everyone for your attention. I want to acknowledge my incredible support from my supervisors and collaborators. Um, yeah, so their guidance made this work possible. Feel free to contact with me on LinkedIn and email. I'm happy to answer any question. Thank you, everyone.